What was that, Oscar?
I'm gonna put that on HD.
Oh yeah, no HD. No, it doesn't record enough. The time, the total time goes down as the the quality increases. I think so. Yeah. Good. Grainy. Good. That's a good thing. Okay. So I want to talk about transcription and translation. These are terms you may or may not be familiar with. If you're not, then that's all right. We're going to learn them. First, I want to get the terminology down because a lot of people maybe have misconceptions about what the genome is versus what a single gene is versus what gene expression is. So your genetic information is basically a cookbook for how to make stuff in your body, right? So this is, you saw this the first week, right? Cooking for 1 or 2. You had cells on the end, and you'd have a good little analogy. So the cookbook would be your genome. So the genome is the complete set of genetic information. One single recipe would be a gene, okay, which is just one sequence of nucleotides. Keep in mind there are 3.2 billion nucleotides in your genetic code, okay? One sequence of nucleotides that codes for one protein to be made as a gene. And the exome is the name given to all of the recipes, okay, all of your protein. Every protein in the body has an instruction or a recipe. That's called your exome. And that makes up only a small portion of your genome, which is weird, which means there's a lot of useless letters in your genome. That's a mystery. So when they mapped the Human Genome Project, if you guys have heard of this, it was, I think, in the early 2000s where they mapped the entire human genome. And they found out that there's like 99% of that is junk DNA. They called it junk DNA, meaning they don't know what all these little letters stand for, but they think that the junk DNA is nucleotide sequences that control gene expression. So they think the seemingly random sequences control the exome, which means what proteins are created at what times. So gene expression is turning on and off protein creation. So sometimes you turn it up, sometimes you ramp up protein production, sometimes you ramp it down. So in a drawing, this is sort of what it looks like. I'll keep flipping back. So one chromosome is basically a packed-in version of one DNA strand here. So if we talk about DNA strands, a DNA strand is in the shape of a double helix. If you look really carefully at all license plates, you'll see a hologram of a double helix right in the center. I don't know if you've ever noticed that. I didn't really notice it until recently, but it's kind of interesting. And as you wind this DNA strand up around little proteins called histones, you compact it into a single chromosome. How many chromosomes do we have? 23 pairs, so technically 46. Those individual nucleotides are built from a phosphate. I'm sorry. Yes, a phosphate, a sugar, and a base. There are 4 different nucleotides that you use. Every nucleotide has a standard phosphate and sugar. They all look the same, but the base differs. What are the different bases? Adenine, cytosine, guanine, and thymine. So A, T, C, and G. Chargaff's rule states that C always pairs with G, and A always pairs with T. So they're always going to be on opposite sides of the chain. DNA resembles a ladder twisted into a double helix, as we said earlier. When you hear backbone, it's the sugar and phosphate that are considered the backbone that kind of hold it together. And remember when we saw phosphates on ATP? When we said they give they have an output of some energy for our body. The reason DNA has a phosphate group is because it powers its own replication process. So it needs a little bit of a fuel there. So think of it as kind of like a reserve fuel tank. Here's RNA, which is single-stranded molecule, resembles DNA, which is a double-stranded molecule. If you're having trouble remembering which one's double and single-stranded, double-stranded, D. And what are these, what type of bond holds those bases together? Hydrogen bonds, good. Okay, so I think that's all we need in terms of introductory information for DNA. Now we're going to talk about the processes in which we access the DNA. So there are two reasons we access DNA. One reason is to perform mitosis. Okay, that's also known as replication. When do we do that? When do we perform replication? Yes, Green.
G1 phase? No, G2 phase.
What's in between those two?
Oh,
S phase. Good. So S phase, S stands for synthesis. What are we synthesizing? We're synthesizing DNA. So remember that a large majority or chunk of a cell's life is spent in that S phase, which is part of a larger phase called interphase. And that's where we're doing this replication. The second reason we're accessing the DNA, so we're only accessing the DNA for replication in one part of that cell's life. In the entire cell cycle, we are accessing the DNA constantly to make protein. So let's talk first about replication. Replication is involving 4 steps, just like excitation and contraction, right? 4 steps of symmetry. This is some nice symmetry here. DNA replication, first, before we can replicate it, those hydrogen bonds have to break. There is, anytime bonds break, there's probably an enzyme doing that, right? And the enzyme, I don't know if it's listed, but if you wanted to know, it's called helicase, because we have a double helix. So that's enzyme that snips up the double helix right in the center, snips the hydrogen bonds, is called helicase. That allows the strands to unwind and separate. So have you ever like pulled apart a rope? So your rope has like generally like a few strands. When you pull it apart, it kind of twists on itself while you're pulling it apart. So just keep in mind whenever you do this, whenever you're pulling it apart, you have to also straighten out those strands so they don't twist around each other. And there are other enzymes that do that. Nucleotides pair, new nucleotides pair with one side of that DNA. So the exposed bases on one side will be unzipped. We snipped all those bonds. We exposed both sides of the DNA. On one of those sides, we take that and use it as a template to make what? I'm talking about RNA right now. What am I doing? Okay, so we're doing both sides here. Autopilot. Okay, so DNA unwinds. Then we've got 2 single strands. All right, DNA polymerase, an enzyme, comes in and accesses both of those strands and adds new nucleotides that are complements to each individual base. So it's a pretty simple process, really. We just take those 2 exposed sides and we add on from our storage. We have a storage, like a toy box in the cell with all these spare nucleotides, and we just tack them on to one side. So we end up with 2 daughter strands of DNA from one mother strand. It's just like mitosis, but with DNA, right? So on a smaller scale. Remember that this is done during this S phase here. As every chromosome has to be copied, guys, so this is just one DNA strand from one chromosome. You have to do this with all the chromosomes, which is why when you look under the microscope at a cell that's in prophase, it's really, the nucleus is like really, really dense. It looks like this dense bowl of spaghetti. That's because we literally doubled the DNA. And then it can undergo mitosis. So whenever that DNA polymerase enzyme is adding on nucleotides to the daughter strands, there are other enzymes that come in that I suppose we don't have to know the names of, that add in, that kind of seal off and glue together that sugar phosphate backbone that we were talking about. So you need to solidify the molecule, which is that ribbon here. That's indicating the sugar and phosphate backbone. So replication is pretty straightforward, I would say, but transcription and translation are just a bit more complex. So we spend a little bit more time on protein synthesis. So how do we take this DNA and create a protein using the instructions? There are two major steps to that. The first one is transcription, the second one is translation. And we use what's called the triplet code, meaning every 3 nucleotides codes for 1 amino acid. Okay, every 3 nucleotides in that gene sequence for that recipe, for that protein, codes for addition of a single amino acid. So therefore, the sequence of bases in that little strand of nucleotides is determining the amino acid sequence in a growing polypeptide. Polypeptide is just a long strand of amino acids. So DNA stores the what's called the master copy of genetic code and remains in the nucleus. So hopefully that code never gets altered, because if the code gets altered, then we don't make proper proteins anymore because the sequence has changed. And that's what's called a mutation. Sometimes you're born with those mutations, the altered DNA, which is what causes sickle cell. Sometimes they happen later on in life. Yeah?
Okay. That's the tricky part.
Right.
When the DNA is first being replicated and it's first being separated and you're adding this, I guess, Okazaki fragments.
Oh, yeah. So this is a little more detailed. We get into, but that's okay.
I'm just trying to visualize it.
That's all right.
When there's a mistake made in that step, it's easier to correct. But then after that step, like we were talking about, if there's a mistake in the later phases, like during interphase and, not interphase, I'm sorry, translation and transcription, if the mistake hasn't been corrected at that point, then that protein is going to be deformed. There's not going to be any repairing DNA or any repairing enzymes at that point.
Yes.
Okay.
So if the master code is altered, then you're going to have issues making all of the proteins from then forward in that cell. But if just the addition of those amino acids is altered for one of those gene sequences, it's not retroactive, meaning the DNA hasn't been altered, it's just later on we had, a mistake with making the protein. The next protein that's made, since the master code stayed intact, is probably going to be made properly. Does that make sense?
So, okay, we're going to get more complicated.
Probably.
Because I'm thinking like, let's say in the process, there's an error, like it just, maybe the DNA polymerase 3 just simply made a mistake. And then, as the second protein is coming along and looking at it, it says, oh, that's a mistake, A is supposed to be your T. If that happens before translation, then I guess everything's okay, but then, let's say.
No, if that happens before translation, then when translation occurs, there's going to be every protein that's made is going to be mutated. Because what you just described as the master code has been altered.
Okay, so if the master code is altered, like let's say we're talking about the synthesis stage in the cell, so
Right.
that's messed up. Then all the other parts are going to be a domino effect.
Correct.
But if it happens in translation or transcription, then
In copying the code, then
it'll be fixed.
it'll be fine.
That's the complicated part, because
Right.
it seems like, it just seems complicated how that error can be corrected in one stage, but then it can't be corrected in
Right.
the first stage.
So basically, the error is made during the S phase when we're replicating that DNA. Then there's no fixing that after interphase is over. Okay.
Sorry.
Did that answer your question?
Yes, yes.
Okay, good. All right. So, in what form do we copy the DNA during protein synthesis? We copy it into a messenger called RNA. So, when we're making proteins, we don't shuttle the DNA out of the nucleus. It never leaves. We just simply make a copy of one side of the DNA, and then we send it to a ribosome. And remember that ribosomes make protein. So that would just be like, if you've got the cookie and you want to make mac and cheese, whichever, you definitely want to make mac and cheese. Mac and cheese represents a protein. You're not going to cut out this from the book and put it somewhere in the kitchen or give it to somebody else. You're going to make a copy on an index card. The index card would be the RNA, and then you give away the recipe to somebody. Otherwise, then you don't have that recipe anymore, right? So you cut out part of your genome, which you don't want to do. Mini meatloaf. Looks good. Okay, so 2 parts to protein synthesis: transcription, translation. I always had trouble remembering which one came first. Transcription comes first. How do you know that? You look at the words, transcription and translation.
C comes before L.
Yes, very good. Transcription, translation. C comes before L in the alphabet. Transcription comes before translation.
And kind of like what she was saying, if things get lost in translation, things can't get fixed very well.
Lost in translation.
Yeah, in the movie with Bill Murray.
Yeah, it's a good. I love that. Right. So translation. Something gets lost in translation, then the protein will be altered.
Yeah.
Okay. Messenger RNA or mRNA is the name given to the molecule that's made. I did this wrong. Well, bear with me here, guys. Okay, so DNA is transcribed to mRNA. This is my little flow chart I use. Okay. And we'll talk about translation in a moment. For now, we're talking about DNA being copied or transcribed. So maybe you could use the C for like copied. DNA is getting copied into mRNA, but all we're doing is we're only copying one side. That is done by an enzyme called RNA polymerase, which makes sense because we're not making DNA from DNA, we're making RNA from DNA. So this enzyme is responsible for adding in the proper amino acids to that for adding the proper nucleotide bases in that RNA strand. Here's the one thing that's different about RNA. When RNA is being made, every time you would substitute or complement a T, we always throw in a U. U is always substituted for T. It's called uracil. And in RNA uses uracil in place of thymine. So just remember that uracil is used in place of thymine for RNA. Every time you were going to add a thymine onto that strand, you add a uracil. Okay, the steps in transcription are as follows. RNA polymerase recognizes the proper strand of DNA first, and then as soon as that gene is exposed, I'm going to go back to my cookbook. as soon as, so I would be RNA polymerase, I would open up the cookbook, I would expose sweet potato puffs. Awesome, that sounds good. Sweet potato puffs, okay. As soon as I've exposed sweet potato puffs, I only copy that recipe, so I know what recipe to copy. I'm making a complementary strand to DNA. Okay, I'm using uracil. Every time I would have put thymine on there, I'm placing uracil down on that growing strand, and I'm terminating my copying as soon as I've seen the last step. As soon as it tells me how many calories I'm eating, right? That's when I terminate creation of that recipe or copying that recipe. So the mRNA strand is released. My index card, right, is released. The DNA rewinds into its double helix. Okay, so we're closing the book back up. The mRNA, once it's created, so I wish I had an index card. We could get a visual. Okay, this is fine. So this is a huge index card. This is my copy. Okay, so as soon as I've copied that recipe, I'm folding it up and I'm throwing it through a nuclear pore because I want to get it out of the nucleus. The mRNA leaves through the nucleus and latches onto a ribosome. Okay, so visual, right? Here's our RNA strand that's been created from DNA. Remember, instead of adding a T here, which normally complements A, we're adding a U. Okay. Remember that every sequence of 3 bases is called a codon. A codon is an appropriate name because it's coding for an amino acid. Protein synthesis actually occurs in the cytoplasm, which is why we have to get our index card out of the nucleus. It's happening at a ribosome, so that mRNA with the index card is leaving the nucleus and binding to a ribosome. Remember our rubber chicken? The rubber chicken was the prop we used to represent a ribosome. Rubber chicken laying eggs, ribosome laying protein. We latch that mRNA onto a ribosome, and that ribosome will read the code and spit out a protein. That's called translation. So translation is reading this RNA code and spitting out protein. Remember that every 3 bases in that RNA codes for 1 amino acid. As soon as we're done adding all the amino acids we need, we cut that signal off. So all together, this entire process, you want to label this as protein synthesis. Make sure you do that. We're representing protein synthesis with this diagram. I think this is a great diagram. It's a little busy, but I like the visual that it portrays here, that it gives. So when we're looking at this, we're seeing the nucleus here. So we're seeing the DNA that's been kind of ripped apart, kind of like a zipper, right? If the zipper on your gym bag stops working every time you try to open it up, it just rips apart and it doesn't work right. That's what I imagine here, with the teeth being the nucleotides. So we ripped apart the DNA only in the area where we needed the gene. Okay, only in that area. And there are what are called promoters here that promote that to happen, little proteins in the DNA. And we copied one side. This copy is called what, specifically? mRNA. Does anybody know what M stands for?
Messenger.
Messenger. Okay. Good. Way ahead of me. Okay, so we've got a messenger RNA leaving through the pore, latching onto a nucleus. What? Latching onto a ribosome. What did I just say? Latching onto a ribosome. We have a top portion and a bottom portion. It's like a hamburger. Ribosomes are made from 2 subunits. Okay? It's a tasty, like we just had 4th of July cookouts, right? It's a tasty hamburger.
It's like hamburger.
Hamburger. Good one. Yeah.
That's the purple guy, right?
Yeah. Wait.
No,
Grimace.
that's Grimace.
Never mind,
There
that
you
joke
go.
doesn't work.
Sorry.
Glass.
Hey, I mean, I'm always trying to. You just gotta keep trying until you get it right. Remember, you always toast your bun, though. Okay, always toast your ribosome before you latch an mRNA onto it. Okay, so the bun comes together. The top part, which is a little bit bigger too than the bottom part, it's more flat on the bun, right? And then we send the mRNA strand through it, kind of like those toys that you probably used to get at McDonald's, right, in your Happy Meals. Remember those toys with like the little teeth? It was like a car, and then you had this like sort of strand of teeth, and you pulled the teeth through the car, and the car shot forward.
Yeah.
Love those toys, right? That's kind of what I picture when I picture the teeth of the mRNA shooting through the ribosome. As every 3 teeth are read, 1 amino acid is brought in. There is another molecule that's specifically responsible for going and grabbing the right amino acid and bringing it to that ribosome. It's called tRNA, or TAX stands for taxi-RNA. Just kidding. Stands for transfer. Stands for transfer RNA, but I want to call it taxi-RNA. So write that down if that helps. Transfer RNA, depending on what triplet is read, adds in the right amino acid. Once the strand is finished going through the ribosome, then that amino acid chain is cut off and you have a final protein. Remember though that polypeptide chain is just a primary structure of protein. It's just a string of amino acids. Then what has to happen to it? Yeah, so it's going to have to, even in simpler terms, it's going to have to be folded up into the proper shape in its final form. The Golgi is participating in that process. The Golgi will help fold it up into its final form and then export it to wherever it needs to go. Okay, is that good? Does that help? Help you understand translation, transcription. Use this flow chart and then fill in the details from there. Yes,
Okay.
Green, sure.
On the exam, are we gonna have to know?
Memorize the triplet code?
No,
Is that what you're
no,
gonna ask? Yes! No, you're not.
I'm thinking about, because in the book, I think it shows it, how the DNA is only translated from the, I guess we call it the primary end, the promoter end, or the, there's another term for it. But anyways, it only goes from one strand, and that other strand just kind of sits there. Are we gonna have to know the 5' end or the 3' end?
Oh, no.
No, okay.
No. In a biology class you would, but not in an anatomy class.
Just wanted to know.
Oh, so you're talking about DNA is copied from 5' to 3', and then during yeah, forget about that. Okay. Okay, so tRNA brings in the amino acid. What it also brings in is an anticodon. So guys, so if my 3 fingers represent the 3 nucleotides on the mRNA strand, okay, that tRNA strand, if my thumb represents the amino acid that it's bringing in, it's also bringing in what's called an anticodon, which is basically like a fact checker. It checks those 3 codons, puts on a complementary 3 codons to make sure that it's brought in the right tRNA. It's like a spell check, sort of. Those 3 codons are like a little spell check, and then that allows the proper addition of the proper amino acid. That's called an anticodon, that spell checker, and it pairs up with that codon on the mRNA strand. So as the ribosome moves down the mRNA strand, each tRNA brings in its proper amino acid to be added to that growing protein. When the stop codon comes in, then that amino acid is cut off and it starts to fold up into its final protein. There's always a start codon. It's AUG. I always remember this because My life started in August. No, I have an August birthday. Or school starts in August, right? So AUG. Huh?
Boo.
Yeah, I know. School reference. Yeah, gotcha. So AUG is our start codon, and it actually codes for the addition of a methionine onto the strand. So if you look at every single protein that's made, the first codon in the strand is always methionine, because that's the amino acid that's coded for from AUG. Yeah. Go ahead.
Okay, so in translation, like you have the mRNA codon, kind of like what you said, and then if the tRNA comes in and there's like a typo in the anticodon, but it attaches anyway, like would it still attach even if it's the wrong anticodon?
Yes, so every mutation is possible. Like
Okay.
anytime a mistake can be made, it could be made, if that
So,
makes any sense.
yeah.
Yeah.
Okay, so if there's like a typo going with your reference and it kicks it off, that protein is pretty much different.
Done. Yeah.
Yeah.
Because every single amino acid has its job in the
Mhm.
folding process. So if you've got one mistake, the entire folding process from there on out is going to be messed up. Yeah. Cool. Does that answer your question pretty well? Yeah, and a mistake can be made anywhere. It can be made there, it can be made during translation, it can be made during transcription. Fortunately, we have repair mechanisms that actually check, again, double check and triple check to make sure everything happened properly. Okay, this is the triplet code. You gotta memorize this. Oh, I'm kidding. Actually awful. So, you do have to remember that AUG is the start codon, and it codes for methionine. And you've got three, it's also important to note that you have three different stop codons. You have three different stop codons, but you only have one start codon. Okay? The stop codons don't code for any amino acid. They just say it's time to cut off that last amino acid that was added. Okay? Whereas the start codon does code for methionine. Most amino acids have 2 to 4 mRNA codons associated with their addition. Meaning, arginine, which is one specific amino acid, actually has four different codons associated with it. And if any of those codons get encountered, arginine is going to be added. Whereas lysine only has 2 amino acids associated with it. Why? I have no idea. I don't know, this is just the way that things turned out. Maybe you use arginine more often than you do, lysine. But that you could hypothesize about that and make up your own rationale there. What do you guys think? Why would we have 3 stop codons and only 1 start codon? What do you think the reason for that is? I always like to think about why things are the way they are in the body. Let's take it a step further. Yeah.
I know for bacteria that's helpful. The RNA having multiple codons.
Oh, why is that?
Yeah, it's helpful for bacteria because bacteria encounters like a bacteriophage, and the bacteriophage is an RNA bacteriophage. It can attack the bacteria's RNA because the thing about viruses, they can attack DNA, they have DNA, or RNA, they have RNA. So bacteria not having a nucleus, its only defense mechanism is having enzymes and very unique ways of manipulating or having a more flaccid RNA or DNA in order to do the same things that we would do, but with not all the extra parts.
Oh, so they kind of cut off their own production to save their own lives. So it'll help have more stuff. That's interesting.
Yeah, because they have something called endonuclease. Endonuclease, and it's a protein, it's an enzyme that will look for basically like an issue with the RNA or DNA, and if it catches it, it will splice it off and basically segregate it from the rest of the DNA and RNA or RNA, and then that would be at some point it's some point it will just leave the cell, sometimes by exocytosis.
Okay.
Sometimes it gets carried to another bacteria. It's very odd how it
Interesting.
happens.
So it's like apoptosis of the genetic code. Cool. Okay. Anybody have any other ideas of why that might be? Yeah, go ahead, Jim.
Like, is it easier to get it started than slowing it down, maybe?
Yeah. Well, but if you only have one start codon, I don't know if you would say easier to get started, but you
Kinda
always
like
know you're going to
starting
start
top
at the
of
right.
hill, like, you
Yeah.
go down and it's harder to stop versus just being on a flat plane.
Yeah, that makes sense. Yeah, you always know you're going to start at the right spot too. If you've just got one start codon. Sure. What else do you guys think? Any thoughts on this? And this is kind of cool, right? Yeah.
I mean, kind of like what Sabrina was saying, the part of the reason why there could be 3 stop codons is to prevent mistakes.
Uh-huh.
Because if it notices like a mistake coming, it could stop and then start again with a new sequence.
Yeah, that makes sense, right? So we can cut it off, once again, cut it off before it becomes a problem.
Yeah.
Interesting. Yeah, awesome.
Maybe, I don't know, there's no point to this, it's just kind of like a rant.
Okay.
Just like the idea of evolution, how, you know, the things that you don't really need will eventually your body, or the idea of evolution is you just don't, you'll get rid of them, right? So you can start using the things that you do need slowly, so maybe the code at some point did have many starts, and then didn't find a reason to get rid of the stops.
Yeah. So not necessarily like natural selection, but natural progression,
Yeah.
sort of. Yeah. Cool. Good stuff, guys. Anything else? Yeah, I just think this is kind of weird. It's cool though. Yeah.
On a side note, bacteria and, prokaryotic cells and eukaryotic cells, bacteria and prokaryotes, tend to have the same start codon,
Mm-hmm.
which is very interesting.
Yeah, and it's important to also note that the triplet code, the triplet code system is used in all organisms. Okay, so in all organisms, whether you're human or not, you know, this system of triplets is used, which is kind of cool, which means it's really efficient. Yeah. okay.
One more thing, that same start codon, it's the same codon pattern,
Uh-huh.
but for eukaryotes, it's slightly different, which is odd. It's the same protein, but it's slightly different, like by just a small smidgen. It makes such a big difference.
Yeah, any little tiny mutation can change the whole thing, yeah. Okay, so this is just that process visualized, adding in new amino acids here, just in a little bit more detail. So you're welcome to kind of look through that. Like I said, we're not gonna have you memorize every codon, its associated amino acid, but we want you to know this process and how it works. So you should be able to work through if the DNA has this code, right? CTC, okay? You should be able to work through, okay, the RNA would have GCG, okay? And then the amino acid would be associated with that GCG. Does that make sense? Okay, so you should be able to work through how this works. The best way to do that is just to practice one time. So here are those steps in order. They're actually a little bit more expanded here to give a little bit more detail. But let's do something real quick. Let's do a little practice sesh. Let's do some what?
No.
All right, so if AUG is the start codon, and AUG, remember, would be mRNA, so you can kind of work backwards. What would the associated DNA code be? Good. A little louder, Erica.
TAC.
TAC. Good. Now just give me a random sequence of DNA bases now. Go ahead, Lauren.
Cat.
Cat? That's a good one.
Words with friends.
We're just friends. seen Gattaca?
Yeah.
Gattaca is all nucleotide bases. Probably not.
I was gonna say you missing an A, Gattaca.
Gattaca.
Oh yeah, okay.
Don't question me. Just kidding. No. Just kidding.
That's all film.
Huh?
It's all film.
Oh, it is on film. Shoot, man. Someone's gonna fire me. Better not post this online. Alright, so what would be the associated RNA bases here? So this G pairs with C. What pairs with A?
U.
U. Good job. Didn't fool you. What pairs with T?
A.
A. What pairs with G?
C. A. U.
U. Good.
G.
G. U. Alright. Our triplets. Waka waka. Guac. Nice. That's one of my favorite foods. Uh-oh. We got another AUG here, so that doesn't work. So we're going to cut off the amino acid strand here. Sorry, Gattaca. UGWTAC. Okay. All right. So make that protein using that chart, the triplet code in your book. So the first amino acid would be what? Thionine. The second one would be G-way. Yeah, so G, U,
It's at the bottom of the
It's
line.
Valley.
U-A. Okay, I'll take your word for it. I can't find it.
Don't question me.
Nice. Good job, Joe. Okay, what's next? What is it?
Leucine.
Leucine. L-E-U. Okay. What kind of bonds are these connecting the amino acids? Peptide. Peptide bonds hold together the primary protein structure. Hydrogen bonds hold together the folded protein structure. So once it starts to fold, that is hydrogen bonds. So this is primary protein structure, and that is protein synthesis in action. Okay, so from this, words to actions. Okay, because what are words without actions?
Just
This
words.
is anatomy class.
Okay. All right, we're done. You guys have any questions?
I do.
Okay.
Okay. I get where the mRNA comes from, but where does the tRNA come from? It's like it just appears.
Yeah.
Does it disappear?
Yeah, it's stored in the cell.
Okay.
Remember where? So we can talk about its origins though. So remember in the nucleus, there's this little tiny bundle of stuff. What's the darker region in the nucleus? Nucleolus. That contains RNA parts. So we're talking about like any RNA that you have, whether it's mRNA, tRNA, rRNA. Believe it or not, the ribosome is also made of RNA. Any RNA structure generally comes from inside the nucleus. So that's where it was created. But then once it's made, it just kind of hangs out in the cytoplasm and waits for this to happen. And when it's called in, it goes and it grabs the amino acid and it brings it to the ribosome.
Okay.
So like it's on call. Cool. Any questions?
Mini hospital.
Yeah. It's just, it's an operation. We've got all these different molecules coming in, all the tRNAs are getting called in, right? Got the Uber app on your phone. ribosomes like texting tRNA, come get me at Kilroy's. Questions? Alright. I think we're gonna be done. You guys study up for the lab quiz tomorrow. Don't forget we have a lab quiz tomorrow.
And then an exam on Tuesday.
Okay, so we have a lab quiz tomorrow. Exercises 19 through 23 for muscular system. You had labeled, hopefully by this point, all the muscles in those activities. What we do for the lab quiz is we just put up images of those muscles, and you have to identify them. For the lecture exam is on Tuesday. You have the whole weekend to study chapter assessment questions. Chapter assessment questions over and over and over and over. Okay, Learn Smart Recharge. You guys know how to recharge on Learn Smart? It's a very helpful tool. Okay? Have a great day.
We don't have to know for
Mutations won't be on the exam, but it's interesting stuff. So feel free to read through it. There's some cool case studies. Yeah, bless you. You're welcome.
Yeah.
I'll be right there.